structure, okay, the structure. So if you see the structure, you have to study this one. Definitely MSc class 1 molecule and class 2 molecule, how they look. And there are few points which are very simple points that describes how it is being comprised of. Comparative analysis also you can see. And the functions, when we talk about the function, the same picture, I think you have studied, I told you during the APC, who is recording this session? Okay, so this, yeah, so this function as how the antigen is being presented by the APCs, that is only the function of MHC molecule. Now, this is the all MSc structures. So the basic thing you can see that in a MSc class 1 molecule, it is alpha 1 and alpha 2, which comprises the peptide binding cleft. This means this is the area where the agrotope of the antigen will be bound and being presented to the T cells. And here a short beta 2 microglobulin will be there, which is actually a kind of a hallmark of any nucleated cells. Any nucleated cells, they will have this beta 2 microglobulin. So if you are taking some, am I audible?
Yes, 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 ma'am.
So this beta microglobulin function is like that. It's a very conserved protein. And if you check human cells, you will be always taking this as a kind of a hallmark of the nucleated cells in human being. So these two structure and the functions which are very important for you in MHC class 1. This is a kind of a, this is the MHC class 2 function. You can see that this when the endogenous molecules which are coming inside and how by the lysosomal degradation it will be degraded. The same time when this lysosomal degradation is happening, here the from the nucleus the MSc class 1 class 2 molecule will be formed. Now if you go to the back to the structure of MSc class 2, if you see this structure, where is that? Yes, when we see this MSc class 2 structure, this is the class 2 structure. You can see that this completely alpha one chain and beta another full chain is there. So this peptide binding cleft which is being formed in MSc class 1, it was alpha 1, alpha 2. Here it is beta alpha 1, beta 1. So this peptide binding cleft is the major one which will hold the antigenic agrotope for the presentation to the T cells. Now here what I was trying to tell you that time also, that when this is going on, simultaneously the nucleus will be also producing the mRNA, producing the mRNAs for the MHC class 2 molecule, like alpha and beta chain has been formed. So when the alpha and beta chain, you can see that these are the ribosomes, they have been formed and they will come for the protein post-translational modification in the endoplasmic reticulum. Here this what I can tell, the invariable chain of that will be produced in this by the same nucleus at the same time, and this will almost like a temporary blockage of the peptide binding cleft so that the other antigens cannot bind here, or rather I can tell that it is to prevent the self-reactive T cells also at some point of time. When they come here, when they are coming to the Golgi apparatus to form the forming vesicle or the exocytic vesicles, you can see that this part invariable chain which is there, which was almost like occupying the peptide binding cleft, that will be also cleaved. And another protein like the actual antigen which has come inside the cell from the external source, that will now go and occupy onto the peptide binding cleft as and then that is getting presented on the APC. So this is the MSc class 2 function. And if you see that same thing I told you, that Th1 and Th2 T cells which will be upgraded by the MSc class 2 and MSc class 1 definitely TC. These are all the same thing which has been shown. So this is MSc class 1 is inside out pathway, that means endogenous antigens which will be presented, and MSc class 2 is outside in, that means exogenous antigens which will be coming inside to this one. So there is a difference basically. Mm. So this picture is a comparative statement of the exogenous antigens for the MSc class 2 and the endogenous antigens like viral DNA for the class 1. And like this, the pathway will be there. You can see it is very easy to understand. So at the same time, the endogenous, exogenous peptides or the antigens which are coming inside the cell, they will be modified. They will be processed and by the lysosomal enzymes and all. By that time, MSc class, MSc molecule also will be synthesized. So it's a clear-cut difference. You can go through this, and these are all. So this is one thing I just wanted to tell you, that these are the this is the HLA or the MHC gene on the chromosome 6 short arm. It is present between the 6.21. So this is the region. Now this region, there are actually few segments like antibody gene. There are few segments like heavy chains, light chains, and then in case of light chains there are VJ. In case of heavy chain there are VDJ. All those things you have seen. So there are different types of V's. There are different types of J's, there are different types of D's. When they combine with each other in a different combination, you are getting varieties. So here also the same concept. The case, as I was mentioning you, that in case of class 2, there is a something like class 3 whose existence is not very clear. It is very confusing, but there is a segment which separates class 2 and the class 1 segments. Class 2 is mainly DP, DM, DQ and DR. So if you are talking about HLA, again I am telling HLA stands for human leukocyte antigen, because it was diagnosed or found out as a surface molecule on the human leukocytes. Those are nucleated cells. Leukocytes are all nucleated cells. All the 5 WBCs are called the leukocytes. So they are nucleated. So you will get this MSc molecule. Actually, MSc was first diagnosed on the human leukocyte. That's why the name has been given. Now, if A and B, A is coming, here class 1, it is A type. Class 2, maybe it may be DQ type. DQ1, DQ2, DQ3, like that. Class, again, you may get class B, okay, HLA B type. And then in that human, the class 2 may be different. So in this way, human populations can be subdivided. Now, if you take some cells, if you take lung cells, if you want to do this MSc class typing, you can do it. And that is a very good typing actually. So that's why human leukocyte antigen typing, that means it is MSc class 1 and class 2 typing. If anybody can do that one, you can easily differentiate between any population group or any species, any population group, any races or any it is another type of, like what I can tell, that this typing is a requirement for the transplantation. Because nowadays blood group matching is one thing, that is completely one thing, because that is dependent on some cells which are enucleated. There is no nucleus on the RBC, so that is done in a different way. This is for the nucleated cell. So anybody wants to go for a transplantation, HLA typing is a must. Okay, so that is one thing I just want to tell you. So these are the different genes. This is a kind of a configuration how it looks, almost the same. So you can see human complex 2, 3 and 1. Basically 2 and 1 should be given importance to this one. Likewise, mouse also, they have haplotypes. They are not called as leukocyte antigen or something like that. Their MSc molecules are called, once upon a time, it was discovered as haplotypes. Haplotypes meaning in a single genomic copy, you will be able to see these genes are there in a different combination. Number 1 mice is different from number 2 mice because they are different in the MHC molecule configuration. So this is the major thing which I wanted to let you know. These are this is the gene. You can see that alpha 1, alpha 2, alpha 3. This is for class 1. And then transmembrane protein and the cytosolic protein. And then mRNA will be there, and you are going to get this kind of a thing. Lot of work has been done on this. Even for this one gene for beta chain, one gene for the alpha chain. Simultaneously, these two are expressing, and you are going to get, and this is the peptide binding cleft where the peptide is bound to it. Okay, so rest of the part, this polymorphism, as I told you, the key, you don't have to go in details of these slides, because it tells about different types of typing, different types of HLA or the MSC molecules in different consequences. Now, in case of corona also, people have done HLA typing. Like, every people are not getting down with corona with the same symptoms, same consequence, same effect. So that means the infection of this virus also is dependent on something. What is that something? It is on the MSC molecule, because MSC molecules are the molecules which will present the antigen, whether it is foreign antigen or it will be generated inside the body, it will be presenting to the T cell. So your entire immune system is dependent on the presentation. So the presentation is done by MHC molecule. So that is the major thing in MHC molecule. Any doubt, anything, please? Is there any doubt?
No, ma'am.
Anybody else? No? Regarding MHC, any doubt?
No, ma'am.
Good. Then I'll go to the next, that is techniques. Okay. Tell me if you can see the slide. Alpha 1, alpha 2?
Yes, ma'am.
Yes, ma'am.
See, the major thing you have to keep in mind, that antigen antibody interaction, to study antigen antibody interaction in a qualitative or in a quantitative manner, all these techniques have been evolved. Okay, it is need based, this one. So basically this antigen antibody interactions are non-covalent interaction. That's why that is like very strong interaction. You can see there, this does not lead to irreversible alteration. That means it is very difficult to dissociate, and if you can irreversible, it is like what I can tell, that if you can change some parameters after the antigen, which helps antigen antibody to bind, if you change the parameters, they can be dissociated. So most of the time, the antigen antibody interaction is measured by the binding affinity, by the binding quotient, but to quantitate that what is the level of binding, like how much strong or weaker binding is there, there we usually do the dissociation of this complex. As you know that if more time or more effort is required, more energy is required for dissociating to this complex, then the affinity is too high, and vice versa. So these interactions, the techniques which has been evolved, it is to detect antigen or antibody, diagnose diseases in so many cases, most of the cases nowadays, measure the magnitude of humoral immunity. Why? Because humoral immunity, because most of the time we are dealing with the antibodies. Antibodies are direct significant, direct what I can tell, direct indications of the humoral immunity. Identify molecules of biological and medical interest. So basically, these are the bonds which you can find out in the antigen antibody interaction. It can be hydrogen, ionic, hydrophobic, van der Waals force. Most of them van der Waals forces are there, and See, usually I told you antigen antibody reversible, but you have to find out the the dissociation constant. The forward and backward movement will be there in the reaction, but how to dissociate the molecule, that tells about what is the strength of the binding capacity. Usually these are different types of immunotechniques are there, like agglutination test, double or single diffusion precipitation test, you have done it. Immunoelectrophoresis, you have done it. Western blot, some of you definitely must have done in your molecular biology test. Complement fixation test nowadays nobody does, but once upon a time it was very popular. Immunofluorescent testing is the one which is the most modern techniques nowadays. And immunoassays are there, like RIA, radioimmunoassay, and ELISA. So ELISA, all of you know. RIA, once upon a time, it was very much promising and very much used, especially to detect the hormone levels in the serum. But due to the radioactivity usage, it has lost its value in the diagnostic field, and that has been taken over by ELISA at this moment. So agglutination test, you have done it probably. This hemagglutination test, like how you do bacterial agglutination, passive agglutination, and you can do also one more thing, agglutination inhibition. Just first you give the molecule antigen, and then instead of the antibody binding something else, you can mask them with that one. How much ever it cannot bind, like negative reading will be giving you some positive things. So usually you know that agglutination reactions are happening in two stages. Stage one, that is sensitization, that is attachment of the antibody to the antigen. Okay, so and the second one is lattice formation. So it is like this. First, hello, excuse me. So you can see the first one, sensitization, antibodies are binding with the antigens, and the second stage, which is there, which forms the lattice. Like this is actually agglutination. So this is one test, antigen antibody. This is one example, is your ABO blood grouping kit, what you have used in your life. So these are the bonds. Please go through these bonds. I'm not going directly. This one. So, which are the factors which affect actually the this one? You must have seen some temperature is required. You have kept after SRID or ODD. You have kept the plate in the moist chamber, at 37 degree centigrade for overnight. So these are optimization, optimum temperature, optimum moisture. Everything should be optimum. Otherwise, this binding will not be proper. So temperature actually is one of the major factor. Next is the pH. That's why you are using an assay buffer all the time. This kit will have some assay buffer. You must have seen in that assay buffer only you are dissolving the agarose, and you are making 1x assay buffer. You are dissolving the agarose and you are making the gel out of it. And even the antigens and the antibodies, they are also dissolved in some proper buffer, so that proper buffer to maintain the pH for this one. Incubation time is very important. Something, it can happen 2 hours incubation at room temperature. Sometimes it may require overnight incubation at 37 degrees. So this particular things are very much need standardization, and whatever the kits you have used in the lab, they are all standardized protocol. So then ionic strength, like which, if PBS, you may have to use PBS. PB stands for phosphate buffer plus saline, sodium and chloride ion. Those will give the antigen antibody molecules better strength to bind along with this one. So the actually it has happened that usually we don't increase most of the, we don't increase unnecessary ionic strength in the like in the solution. So less is better for this antigen antibody reaction. It may not be so high. Antigen antibody, they are very reactive substances. So, in case of reactivity means in case of binding, I'm telling, because they are having very high affinity towards each other. So this is the way this even in the agglutination, this is the formation. First few antigens will be blocked by the or bound by the antibodies. After that, there will be some more, and thus the molecules will be like this. So if you just increase more amount of antigens or more amount of antibodies, it may not help. Optimum antibody concentration, antigen concentration is required to get the precipitin band or something like that. These are the factors. Other factors: size of the immunoglobulin, big, small, it can be happen like IgG, it is monomer, IgM is pentamer, IgG will be having 2 binding sites. Here IgM will have 10 binding sites. So that is also important. You can see that these are the different blood antigen molecules. The molecular weight also very important. You have to see that